our total body weight, right? So the blood that flows through our veins and our arteries makes 8% of the total amount of body weight that we have. Now, if we take a sample of our blood and we spin it so that we can break it down into the different components, we find that about 45% of the blood is going to, of whole blood, is going to be made up of these formed elements, or really these cells, right? Then there's this buffy coat and followed by plasma. So at the very bottom, you see the red blood cells. There's a buffy coat that separates or sits right on top, and then all the way on top of the tube, you're going to see plasma. And plasma makes up 55% of our whole blood. So 55% plasma, 45% to blood cells. And the majority of the 45% is really your red blood cells, right? More than 99% of all cell types in your blood is going to be red blood cells. And less than 1% are your white blood cells or your leukocytes, and less than 1% will make up your platelets. When we look at plasma and the 55% that it holds of whole blood, it's made up of water, proteins, and other solutes. And of the different proteins, we have albumins, globulins, fibrinogen, prothrombin, multiple different types of plasma proteins that actually play very important roles. All right, so when we look at blood cells, so all blood cells that we have, they have a limited lifespan, and our body has the ability to continuously produce these cells whenever we need them. Actually, they're on a regulated schedule of release. And they're generated from a common stem cell found in the bone marrow. So really, it's just a single cell can, differentiate and grow in response to multiple different stimuli and become any one of the different blood cell types that we have. Now, what exact cell type it's going to become, it's going to depend on the different type of stimulations that the stem cell is receiving. And we'll talk briefly about that in just a little bit. So the main blood cells are the red blood cells, the white blood cells, which are of multiple different types, and finally platelets, right? Although platelets are not really cells, and the same really can be said about the red blood cells as well. When we take a deeper look at white blood cells and the multiple different types of white blood cells, we can break them down into about 4 major components or categories. You have the granulocytes, the monocytes, the lymphocytes, and the natural killer cells. So the granulocytes, you have your neutrophils, your basophils, and your eosinophils, and you can look here at this table to determine their main functions. We also have monocytes, and the monocytes are going to differentiate into your macrophages later, right, once they become activated. We also have lymphocytes, which are very important for immunity, and we have 2 main types, the B lymphocytes and the T lymphocytes, or the B cells and the T cells, and we also have the natural killer cells.
What's up, Cynthia?
Which are very similar to the T lymphocytes, but they're different. They the way they act and respond are slightly different than the T lymphocytes. So this is what a blood smear would look like under the microscope, and this is how the different cell types would appear, right? And so here you notice you have the red blood cells, and then you have the different types of white blood cells scattered throughout. And then you can see how much, the majority of the slide is red blood cells, but we do have some white blood cells scattered. And you see the apparent nature of the neutrophil, right, which is also called the polymorphonuclear leukocyte, right, because of these multiple nuclei that you see here, or that appearance of multiple nuclei in leukocyte, right. called the polymorphonuclear leukocyte, right, because of these multiple nuclei that you see here, or that appearance of multiple nuclei, and compare that to a lymphocyte or a monocyte, which has that very distinct feature of having a single nucleus. All right, that's our introduction. See you next time.
Students with ADHD save 10 hours studying with this Chrome extension.
But no.
Let's start talking about red blood cells. So red blood cells, or erythrocytes, are the cells that are responsible for oxygen transport and carbon dioxide as well, right? That's really their main goal. They stay within the vascular system. Now, this is important because this is in contrast to white blood cells. White blood cells don't stay confined to the vascular system. Actually, whenever there's an infection, they start escaping and squeezing their way out of the vascular system in order to deal with any pathogens that may be present in the interstitial spaces. Red blood cells, however, stay within the vascular system unless there's some sort of trauma or injury that allows them to escape. So RBCs, they're not really cells, from a technical perspective, because they have no nucleus, they have no endoplasmic reticulum, mitochondria, ribosomes, therefore they can't grow or divide, which also means that we need to be able to continuously produce these cells because they have a very limited lifespan, right? As you see here, they can stay alive for about 120 days. Now, our bodies have a very interesting, simple, but I think it's an elegant system of recycling these red blood cells, and so we'll talk about that. But basically, when they're worn out, as they pass through certain parts in the circulatory system, specifically in the spleen, the healthy red blood cells are able to squeeze their way through, while the old flimsy red blood cells are going to rupture and die. But also, our macrophages, right, are responsible for clearing up, phagocytosing, and digesting the older red blood cells as their function starts to decline. Also, remember, when it comes to the metabolism of the red blood cell, they can only use glucose for energy, and since they don't have any mitochondria, they exclusively generate energy via glycolysis, right? There's no oxidative phosphorylation when it comes to a red blood cell due to the lack of mitochondria. So the main functions of a red blood cell is hemoglobin transport, and hemoglobin is responsible for the transport of oxygen as well as carbon dioxide. So we carry oxygen from the lungs to the tissues. And carbon dioxide in the opposite direction. When it comes to carbon dioxide, the carbonic anhydrase reaction is very important. So carbon dioxide binds to water.
Dave, you're still here.
The enzyme carbonic anhydrase is going to catalyze this reaction, giving us carbonic acid, which is going to dissociate into bicarbonate and protons. This is what happens at the level of the periphery. Once the red blood cell reaches the lungs, the reaction goes in the opposite direction, giving out carbon dioxide, which is going to cross into the alveoli and be expired back to the atmosphere. So another important job of the red blood cells is that they carry carbonic anhydrase, right? The enzyme carbonic anhydrase is going to catalyze this reaction, giving us carbonic acid, mitochondria. So the main functions of a red blood cell is hemoglobin transport, and hemoglobin is responsible for the transport of oxygen as well as carbon dioxide. So we carry oxygen from the lungs to the tissues, and carbon dioxide in the opposite direction. When it comes to carbon dioxide, the carbonic anhydrase reaction is very important. So carbon dioxide binds to water. The enzyme carbonic anhydrase is going to catalyze this reaction, giving us carbonic acid, which is going to dissociate into bicarbonate and protons. This is what happens at the level of the periphery. Once the red blood cell reaches the lungs, the reaction goes in the opposite direction, giving out carbon dioxide, which is going to cross into the alveoli and be expired back to the atmosphere. So another important job of the red blood cells is that they carry carbonic anhydrase, right? And again, that's just very important for carbon dioxide transport. So red blood cells' main job is gas transport, oxygen and carbon dioxide from the lung to the periphery and back. So red blood cells cells' main job is gas transport, oxygen and carbon dioxide from the lung to the periphery and back. The red blood cells, they have that distinct shape, the biconcave disc shape, right? And they're able to actually change this shape a little bit and squeeze through tighter capillaries. However, when the red blood cell becomes a little bit too old, its cell membrane becomes fragile and they can be easily ruptured. And so there's certain parts in our system, again, namely the red pulp of the spleen, where red blood cells kind of have to squeeze their way out and pass through the spleen. So the spleen really works as a filter. And what's interesting is when the spleen is removed, the number of abnormal red blood cells circulating in our bloodstream increases, right? Because that filter is no longer there. So abnormal red blood cells circulating in our bloodstream increases, right? Because that filter is no longer there. So, in terms of the quantity or the red blood cell count, in healthy men, that's somewhere around 5.2 plus minus million cells per microliter, and in women it's slightly lower at 4.7 plus minus 0.3 million cells per microliter. When we're looking at the quantity of hemoglobin in the cells, a red blood cells can hold up to 34 grams of hemoglobin in each deciliter of cells, right? So if you had a full deciliter or 100 mLs of red blood cells, you would have 34 grams of hemoglobin. However, our hematocrit, and remember hematocrit is the percentage of red blood cells to the total blood. Our hematocrit maybe was going to be around 40 to 45%, so we don't have a full ml of blood in this scenario. Looking at the hematocrit about 40 to 45%, we're going to hold an average of about 15 grams of hemoglobin per deciliter when it comes to men, and women slightly lower at 14 grams per deciliter, 14 grams of hemoglobin per deciliter, considering that 1 gram of hemoglobin can hold 1.34 mLs of oxygen. So therefore, when our hemoglobin is fully saturated, men can hold about 20 mLs of oxygen per deciliter, and women can hold about 19 mLs of oxygen per deciliter. So we mentioned that red blood cells, just like all our other blood cells except T lymphocytes, are made in the bone marrow. And early on in the age span, most of our bones are participating in this process, including our long bones. But as we age, the ability of our long bones, like you see here in this figure, our femur and the tibia, their ability to continue producing red blood cells quickly and steeply declines with age. However, as we age, we maintain the ability to produce these cells in these membranous bones, such as the vertebra, the sternum, and our ribs. And that's where we get blood cell production. So we such as the vertebra, the sternum, and our ribs. And that's where we get blood cell production. So we also briefly talked about the stem cell, and we mentioned that a single stem cell, in this case the multipotent hematopoietic stem cell, can give rise to all the different types of red and white blood cells, as we see here. Actually, as few as 5 cells, if injected into a mouse that has defective hemopoiesis, are really enough. Just these 5 cells are enough to reestablish the entire hemopoietic system, which is very remarkable. And as previously mentioned, all cells are developed in the bone marrow except for the T lymphocytes, which are developed in the thymus. And macrophages, they're not developed in the bone marrow because they are derived from the monocytes, which are developed in the bone marrow.
This time focusing this on myself. There it is. That's all I had to do. Now we're in. Right, so the monocyte developed in the bone marrow, but then the monocyte, once it becomes activated, it will differentiate into the macrophage. So
the multipotent hematopoietic stem cell is going to really take one of two directions, right? It's going to differentiate into a common lymphoid precursor cell or a common myeloid precursor cell. And without going into all of these, details, but the common myeloid precursor cell also continues differentiating, and it gives you the erythroblast, which is going to give you the erythrocyte of the red blood cell. It's going to give you the megakaryocyte, and the megakaryocyte is a very large cell from its name, and it starts budding off little pieces, which are going to be your platelets. But then, from the common myeloid precursor, you get the mast cell, the basophil, the eosinophil, the neutrophil, the dendritic cell, and monocytes, which later give you macrophages. The common lymphoid precursor will give you the B cell, the T cell, and you see here, and the thymus, and the natural killer cell. So regulation of hemopoiesis, how do we regulate blood cell formation? And these are regulated through colony-stimulating factors, CSF, which are basically these glycoproteins that are circulating in our body and can act as hormones or act directly in the bone marrow as local mediators. So a very well-known colony-stimulating factor is erythropoietin, or EPO. Which is one of these CSF, or colony-stimulating factors. So EPO is produced by the kidneys, and it leads to an increase in red blood cell production. But we have multiple other ones. We have a granulocyte CSF, a macrophage CSF, and as you see here in the table, it kind of outlines their target cells, where they're produced, and the type of receptor that they activate. EPO regulates RBC production. As you see here from this figure, right, so the main stimulus is if you have a drop in oxygen, right here, if the oxygen levels are dropped, any kind of hypoxia or ischemia or reduction in oxygen delivery stimulates the kidneys to release EPO, right? EPO then goes to the bone marrow, where it stimulates the bone marrow to release more red blood cells. If you have more red blood cells, what that should do is that it should increase the oxygen delivery. Well, when you're increasing oxygen delivery, you're inhibiting the main stimulating factor for EPO, and this is where the cycle breaks, right? The negative feedback happens here because you're taking away that initiating factor, and EPO production stops. So cycle breaks, right? The to release more red blood cells. Regulates RBC production, as you see here. But we have multiple other ones. We have a granulocyte CSF, a macrophage CSF, and as you see here in the table, it kind of outlines their target cells, where they're produced, and the type of receptor that they activate. EPO regulates RBC production, as you see here from this figure, right? So the main stimulus is if you have a drop in oxygen, right here, if the oxygen levels are dropped, any kind of hypoxia or ischemia or reduction in oxygen delivery stimulates the kidneys to release EPO, right? EPO then goes to the bone marrow, where it stimulates the bone marrow to release more red blood cells. If you have more red blood cells, what that should do is that it should increase the oxygen delivery. Well, when you're increasing oxygen delivery, you're inhibiting the main stimulating factor for EPO, and this is where the cycle breaks, right? The negative feedback happens here because you're taking away that initiating factor, and EPO production stops. So people with kidney disease, or when their kidneys are removed, they have anemia, right? And they become susceptible to anemia because they lose EPO production, which comes from the kidney. So when you're dealing with a patient with kidney disease, you need to be aware of the possibility of anemia. And patients with anemia, we're going to talk more about what that can, some of the pathology behind that. But remember, if they have a lack in red blood cells, that means they have a lack in oxygen delivery. So it's very likely that they're going to be very tired, they're going to be weak, they're going to be susceptible to a bunch of different problems, and you need to be aware of that. So kidney disease can affect physical function in multiple ways, and part of that is due to the anemia that comes from lack of EPO production as well as other causes as well. Our tissues also have a way of responding to low levels of oxygen. So the kidneys are going to respond to that by EPO production, but our tissues can also promote angiogenesis, which is growth of new blood vessels, if they sense low oxygen. And how that works is that we have a transcription factor in our cells called hypoxia inducible factor, and if you want to be specific, it's HIF1 alpha, that is going to be activated. So HIF, that's all you need to know. Specific, that we have this, which is growth of new blood vessels, if they sense low oxygen. And how that works is that we have a transcription factor in our cells called hypoxia inducible factor, and if you want to be specific, it's HIF. Alpha, that is going to be activated. So HIF, that's all you need to know. Hypoxia inducible factor becomes activated, and once that happens, it stimulates the cells to release a growth factor called VEGF, vascular endothelial growth factor, which stimulates the endothelium to divide and grow, and you start getting this capillary sprouting over here and growth of new blood vessels. In the context of high oxygen delivery, HIF is reduced, it's not expressed as much, and you don't get VEGF release, right? So that's just another mechanism of how our tissues can respond to changes in oxygen delivery. This is an interesting study showing blood volume adaptations with exercise. You can see pretty quickly after exercise, blood volume increased steeply, and then it kind of plateaued all the way out until 80, continuously monitoring for about 80 days. But the majority of that initial change in blood volume, or that blood volume, or that expansion in blood volume, was actually driven by changes in plasma volume. So you see here, plasma volume increased significantly, and then it started dropping. And that's what drove the increase in blood volume, or the expansion in blood volume. When we're looking at red blood cell count, right, the amount of, or the erythrocyte volume, for the first about 3 weeks, there's almost nothing. And then after about 21 days, you start seeing that increase in erythrocyte volume. So what this slide is showing you is that red blood cell production is a process that requires time. If we increase our demand, we will get EPO stimulation and our bodies will respond, but that is going to require time in order for you actually to see functioning, a significant increase in red blood